Hello, everyone. I'm your People Behind the Science podcast host, Dr. Marie McNeely. And today I am delighted to welcome you to episode 661 of the show. We are going to be speaking with our guest, Dr. Robin Buell. And I'm glad to have you all joining me for our conversation today. If you want to continue meeting fantastic scientists through our show, listeners, you can follow us at PBT Science on Twitter, and I'm at PhD Marie. Now today, Robin is going to share some of her own experiences in her life, her career, and her research. So get ready to meet another one of our brilliant people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest researcher, Dr. Robin Buell. So, Robin, welcome to our show today. How are you?
Great. Good morning to you and your listeners.
Well, thank you. Good morning. And we are excited to chat with you today and to learn more about you and your work. So let me start by giving our listeners just a little bit of information about your background. So listeners, Robin is a Georgia Research Alliance Eminent Scholar Chair in Crop Genomics in the Department of Crop and Soil Sciences and the Center for Applied Genetic Technologies at the University of Georgia. She received her bachelor's degree in biology from the University of Maryland, her master's in plant pathology from Washington State University, and her PhD in biological sciences and molecular biology from Utah State University. Afterwards, she conducted postdoctoral research at Michigan State University and for the Carnegie Institute of Washington at Stanford University. She served on the faculty at Louisiana State University, the Institute for Genomic Research, and at Michigan State University before joining the faculty where she is today at the University of Georgia last year. Now, Robin has been elected as a Fellow of the American Association for the Advancement of Science and the American Society of Plant Biologists. In addition, she was awarded the 2022 McClintock Prize by the Maize Genetics Cooperation Advocacy Committee. In our interview, she shares more about her life and her science. So, Robin, we're looking forward to getting to know you, of course, more as a scientist, but also as a person. So, can you start by telling us what you like to do when you're not doing science?
So, I do gardening. I like to spend time with my two dogs. I have two dogs, Sherman and Mr. Peabody. And I also spend time watching college sports, both college basketball and college football.
Oh, amazing. So what's growing well in the garden these days?
Not much, actually, because we've had a cold frost here after the hard frost date here in Georgia. So I lost a lot of stuff in my garden, and I've had to sort of restart my potatoes, peppers. But my potatoes are doing great.
That's good to hear. At least there's a little silver lining there.
Yeah.
And I know you mentioned you've got two dogs as well. And I saw on your lab webpage, you actually have a highlight for all of the lab pets, which I think is amazing. So can you tell us about the pets?
Yeah, so I have two rescue dogs. One is Mr. Peabody. He's a miniature Australian Shepherd rescued from an Amish puppy mill. And he's 12. And then he needed a friend to keep him company after our previous dog died. So we got a cattle dog mix. And of course, we had to call him Sherman.
I love it. Well, it's great to get a window into your life outside the lab, but you do some amazing things in the lab as well. So how do you describe your research to someone who's not familiar with it?
My lab, we work on plants and we work on plants' DNA. And to a non-scientist, think of it as trying to understand the blueprint of how a plant grows, responds to stress like cold or disease, how it decides to reproduce. So we work on a lot of different plants. We work on crops like corn and potatoes and sweet potatoes. We also work on medicinal plants, plants that produce anti-cancer drugs, and then also plants that are just really sort of cool like basil, oregano, catnip, catmint. We work on a lot of different plants that have interesting properties.
Very cool. And I'm excited to dive into some of the details because, as you alluded to, you've got a wide range of topics covered in your research portfolio, from evolution to circadian clocks and everything in between. So I'm looking forward to chatting about some of your projects. And I know as a scientist, it can be difficult to be motivated sometimes because there are a lot of challenges that you face. So we love sharing motivation with our listeners out there who might be going through their own struggles. So do you have a favorite motivational quote or a saying, Robin, whether it's about science specifically or life in general?
I think like as a biologist, we're inquisitive. And to some extent, essentially as a scientist, you want to have complete knowledge of something. And in this case, it's the process of biology. And now we have a lot of technology that can help us understand biology. So a lot of unanswered questions that we've had for centuries, we can actually start to answer. And I think it's sort of a puzzle, and I like trying to solve a puzzle. And there's a lot of things we don't know about plants because most scientific research funds are spent in human medicine for obvious reasons. And I think trying to understand some basic biology can really help us with agriculture. And so for me, it's solving the puzzle. That's what's, I think, interesting. And technology can help us do that. Having an interesting question, some historical knowledge, and technology, you can really ask and answer some outstanding questions that we've had for a long time in biology.
Phenomenal. Yeah, I think this curiosity is key. And I love this puzzle analogy that you've drawn for us, kind of putting all these different pieces together to understand the big picture of what's going on in plant biology. And I know you've worked with some phenomenal people as a scientist over the years. So when you look back over the course of your career, Robin, are there people who really rise to the top in your mind as mentors or role models or just people who've had a big impact?
When you're young and starting out, having role models and mentors, that's absolutely key. And actually, I started working on plants when I was an undergraduate at the University of Maryland. That was last century. And I needed money, and there was an advertisement for a dishwasher in a plant physiology lab, and I applied, and they hired me. And that was sort of the start of doing research. And I worked for a woman who was a well-known plant physiologist that really was very engaging. She was an excellent role model. And working in her lab with her graduate students and postdocs sort of gave me the motivation and also just the perspective of doing research and then going to graduate school. And that, I think, was the first key mentor that I had. And that was instrumental. And it went on from there, from my PhD advisor to my postdoc advisor. They were all seminal people to guide me to where I got to be today.
Oh, that is phenomenal. I know you've come a long way since those early dishwashing days in undergraduate, but where did this journey begin for you? You mentioned that this kind of falling into a plant lab maybe was a little bit more chance, but where did your interest in science begin, Robin?
So I grew up in the country, very rural Maryland, and we had a lot of dogs, we had cats, we had horses, we had chickens at one point. But I wasn't necessarily interested in plants or agriculture. I really wanted to be a veterinarian. When I was an undergraduate, that was my plan, was I would finish my bachelor's degree and then go to veterinary school. But when I was a sophomore, I was taking a zoology class, and there was a lot of problems with the class. It was what we would call a large enrollment class that wasn't very focused on undergraduates. And at the same time, I had to take a basic botany class, and that instructor was fabulous and made plants really interesting. And then at the same time, that's when I got the job working in a plant physiology lab, and that's when it changed. And then I decided I wanted to work on plants. So it was all one of these accidental choices that I made, and this is where I'm at today, because otherwise I would have probably been a veterinarian.
Oh, that's wonderful. And I love these stories where it's sort of an unexpected twist or turn in your career path. Like when people go to college, as I did, with a very clear idea, I wanted to be a doctor. So I was in the pre-med path. And then something changed for you along the way. And I think these research experiences can have such a big impact on students. So can you talk a little bit about then kind of what guided you to the next steps of your career path?
So it's really sort of an interesting path, because once I decided I wanted to focus on plants and not take a lot of zoology classes, it turns out that a lot of the plant-based classes have smaller enrollments. So you get a much better, I think, undergraduate experience, because University of Maryland is a large enrollment public university. And I took a class from a very exciting plant pathology instructor that made plant pathology seem so exciting. He explained why Britain is a nation of tea drinkers and explained the late blight famine that caused mass immigration to the United States. And all of that sort of got me excited about plant pathology. So then I decided to go to graduate school and get a master's in plant pathology. And so it was all of these things, like one after another, in terms of guiding me to the next step. And when I was getting my master's, I got to meet a person who was doing some interesting work on plant growth promoting bacteria, that you could colonize the plant. That would give the plant a little bit of immunity against disease. So I then went to Utah State and I got my PhD working on a plant growth promoting bacterium. And then while I was getting my PhD, I realized there was this new technique of molecular genetics and working on this model plant called Arabidopsis. And I just said, I need to understand this. And I decided to do a postdoc in Arabidopsis molecular plant microbe interactions. And that's when I went to Michigan State University. I worked on essentially disease resistance in Arabidopsis. And I did that as my postdoc. I went to LSU and I worked on that. And I was interested in how plants resist and tolerate plant pathogens. And again, all just by accident, I was visiting a mentor back at the Carnegie Institution, and he said, Robin, this job would be perfect for you at this place called the Institute for Genomic Research. And he goes, they're looking for somebody with your skills. And this is when genomics was very new. Nobody knew how to do genomics. And my first response was, well, I'm not a quitter, because I didn't want to quit my job at Louisiana State University. And as always, he said, don't think of it as quitting, think of it as a new opportunity. So I went on an interview, and within 5 minutes of being at the Institute for Genomic Research, it was an opportunity of a lifetime. It was absolutely clear. And so they offered me the job. I quit my tenure-track job, and I went to what was known as Tiger, and I started sequencing the Arabidopsis genome and the rice genome, and that sort of led me down the path to where I'm at now, doing plant genome biology.
That is so cool. And you recently moved your lab just last year now to University of Georgia. So what has the experience been like there?
So first, it's really sunny in Georgia compared to Michigan. That's the thing that we all notice. And there's lots of tree pollen here. It's been 2 months of tree pollen, so all the cars are yellow. And we've been told it goes on for another month of tree pollen. But it's been great so far. It's a very different university. The people are very friendly. I've been able to recruit new people to the lab. It's very exciting. So I've really enjoyed it. Been here since August. It's April now. So we're really enjoying it.
That is phenomenal. And I know you've got so many cool research projects going on in the lab. It was like an amazing menu browsing through your lab webpage there. So is there a particular project that you're working on at the moment, Robin, that you are just the most excited about that you can tell us about in more detail?
Out of our current projects right now, the one that I think is most exciting to us is we had been working on this plant called Madagascar periwinkle. It's used to isolate an anti-cancer drug, and we worked on it for quite some time with my close collaborator, Sarah O'Connor. And we've done some genome work. We sequenced the genome. We've helped them find new candidate genes in the biosynthetic pathway. And we've just started doing some new technology with that plant. And now we're able to understand specifics about genes and exactly which cell type they're being expressed in. And so we've been able to get a very high resolution understanding of the biochemistry of this plant and how it produces this anti-cancer drug. And so for us to take some new technology, apply it to a biological question, and now have it at a really, really high resolution, it explains a lot of the questions we've had before and also gives us a lot of new opportunities. And so that's, I think, our most exciting project right now, because this technology can be applied to every plant we work on. And it's been a very exciting development in the lab.
Very cool. Now, with the goal of this particular project, are you trying to, I guess, understand where this compound of interest is produced in the plant, or trying to increase the yield of it? What's the big picture end goal there?
The end goal, of course, would be, right now, the plant doesn't produce very much of it. So it's what we would call very low yield. And it's still harvested directly from the plant. So the end goal would be is that you would want the plant to produce more so that your yield when you harvest it from the plant would be much higher. But right now, currently, all we're trying to understand is exactly which cell types is the compound produced in and how exactly is it being produced and what are what we would call the regulatory mechanisms. That is, if we can understand the fine tuning of how the plant decides how and when and how much to make of this, that's the last piece in the jigsaw puzzle. Like, we may know how the plant makes it, but understanding how the plant regulates that is the key to then turning that on or off, sort of like the dial on radio and how loud it is. If we can understand how the plant's actually tuning its biosynthesis, we can then manipulate that to have the plant produce more of the compound.
Oh, that is so cool. Robin, thank you for walking us through one of these exciting projects in your lab. And we've hinted at some of the challenges, of course, that scientists face. There's a lot of unanswered questions and a lot of hard work that goes into science. And I think a lot of people don't always get a chance to see this other side of science, the struggle, the failure. Often you just get to see the pretty published work at the end. So do you have a story or an experience that you've had of a struggle or a major fail that you can walk us through and tell us how you got through it?
Well, there's always failures, right? I think the main thing I always try to tell people that are in my lab is, okay, the experiment failed. What did we learn from it so that we don't repeat that mistake? I would say the biggest challenge we've had is we've been working on another project where we try to understand how plants produce tubers. So we know potatoes are tubers, they're below ground organ, right? And the plant produces it to give it essentially a seed over winter and then germinate during the spring. And so we've been working with a lot of other plants that produce these tubers. Several of them come from South America, from the Andes. And they're just like potato in that as the summer wanes and it starts to become fall-like conditions and it cools off and the day lengths shorten, they'll start to make their own tubers. So it took us a very, very long time to get some of these plants to make tubers because we wanted to actually harvest the tubers. And it just took us a very long time to get one plant to make these tubers. And another one, we've gotten it to make tubers, but they're above ground, not below ground. That's been a big challenge, trying to mimic the exact environmental conditions in the Andes that triggers the plant to make tubers. And we've come very close on one. Not quite exactly what we would want, but I think this is the best we can get at this point.
Yeah, that is an amazing example. And I think it's funny to hear that it's such a struggle to replicate these real world conditions in the lab, because I think that's something that is experienced across a lot of different fields here in science. So how do you tackle that, or how do you go through the logic and troubleshooting to get to a point where you decide, okay, these are the key factors that are missing?
The first thing is make sure you've explored what we would call the literature. Like, has someone done something like this before, or exactly this before? So you collect that historical record of knowledge, and then you design your experiment. And what's really important in that experimental design is that you have controls, that is positive and negative controls, so that something is working, so you can make a determination like, I didn't make a major problem. And then second of all, very simple things like, when we grow these plants, we're growing them either in a greenhouse or what we would call a growth chamber, where we're controlling the light, the temperature, and that we're monitoring that so that we have a record of what has been happening throughout the experiment, so that we can look back at that and say, no, yeah, the temperature's been fine, they've been watered, that's fine, so there's no problems. And I think those are the key things, is to make sure you have a good experimental design to start with, so that the experiment doesn't come out how you predicted. You have these checkpoints so that you can go back and say, well, this wasn't the cause of the failure. And then that allows you to more quickly troubleshoot what's going on.
Gotcha. So do you ever get to travel to some of these places, for example, the Andes, to see what the conditions are like where these plants are growing natively?
I've worked on potatoes since 1999, and we all know potatoes historically, their center of origin, where they came from, is the Andes, specifically Peru and Bolivia. And so I have been to Peru. I've been to Cusco, where there's a lot of native potatoes, land races we would call, and I've been out to research stations to look at the types of potatoes that native farmers would grow in Peru. And that was very, very exciting.
Oh, that is phenomenal. So now that we're on the subject of travel, do you have a particular favorite place that you've been for science?
Yeah, I've been very fortunate to travel, and I've been to lots of very, very exciting places. And the thing that's probably been the most exciting to me was I've been to Peru, as I just mentioned. And while we were there, we got to visit where the Incans would grow corn in terraces and potato in terraces, and also sort of see how it's still being farmed today at these elevations of, say, 10,000 feet. They'll still be growing potato. They'll actually freeze dry it in place. They'll harvest the potatoes, put it out, and then since it's so cold because of the high elevation, they'll essentially naturally freeze dry the potato so that they'll have food over the winter. And while I was there, I got to visit Machu Picchu and see that as one of the wonders of the world. That was probably the most exciting visit, I think.
That sounds like an amazing experience. And I guess it sounds like there's some parallels between how farming was done way back when and how it's still being done today. And I think that's absolutely remarkable.
Definitely. I mean, I think this terracing to reclaim more land so that you can have a higher agricultural output, right? That's still done in most parts of the world. Not done so much here in the United States because of the rugged West, where there's not as much rainfall, but that's done in South America, and it's done in Asia as well, to just increase the surface area that you're growing.
Well, this sounds like an amazing travel experience. And I know in science, there are a lot of great experiences. We talked about some of the challenges, but I'd love to talk about some of the successes as well. I know, Robin, you've had many successes to celebrate. And I think it's important to take a moment to enjoy that moment, because I think the successes can be few and far between relative to the challenges. So do you have a favorite scientific victory, whether it was a big win or even just a small but particularly meaningful one that you'd like to share with us?
I would say there was two things that maybe I don't take a lot of personal credit for, but they were major achievements. And first of all, I was involved in sequencing the rice genome, and that started in 1999. And it was the second plant that had its genome generated. And what that enabled was for hundreds and thousands of researchers that work on rice to actually start to be able to make accelerated progress in rice genetics and rice breeding to make improved rice varieties. So I've been working on rice now for over 20 years. We have a resource that's online, a database that people still use, that people still find to be very, very helpful in their own research. And I think the amplification of that work, even though we finished in 2005, and then we continue to improve what we call the annotation, that is the decoration of what is in the genome and what does it do. We did that until 2011. So over 10 years ago, we sort of stopped improving on that research. But that really has enabled lots and lots and lots of researchers to do very exciting things with rice and improve its production potential, understand disease resistance. And so that's a huge achievement that maybe I'm not directly associated with it right now, but at the time, it was what we would call paradigm changing. It really changed the way people work on rice. And half the people in the world get most of their calories from rice every day.
Definitely. And this is a really exciting achievement. And as you alluded to there, some of these scientific projects span years, sometimes even decades. So how do you, I guess, celebrate a big win like this? Or do you kind of break it up into smaller milestones?
Smaller milestones, because at the time that we were working on it, the technology is not nearly as advanced as it is now. So it took us 5-6 years to do the rice genome, and I could do it today in a week for a fraction of the cost, and I could do it better. So it was long slog while we did it, and to see now that I could do 100 rice genomes in 6 months and do them all better, that's just amazing. And so you sort of do distance yourself from all the work you did, because now the technology has advanced so much. And sequencing a rice genome would be seen somewhat as trivial right now, because the technology is so much advanced and it's inexpensive, and the tools to do it are what we call open source. Anybody can use those. And at the time, it was very difficult to do it, very expensive to do it. And so to me, that removes a lot of the work that we did in terms of like how much effort it was and how many people contributed to it. It wasn't just myself. So I think it's important to look back and say early pioneers, which I was an early pioneer in doing plant genomics. It was definitely a lot of work, and the rewards were great.
Definitely. It's amazing how far we've come so quickly. And you mentioned that there were 2 maybe remarkable experiences or successes that you've had. Was there a second one you'd like to share?
Yeah, so in addition to rice, I've also worked on potatoes since 1999. And I was involved in generating the sequence of the potato genome. And again, that has been very transformative. And so even though potato is a really important food crop across the world, there's very few people that actually work on potato in terms of breeding new potatoes or doing potato biology. And potato is also a more complicated genome. It's much bigger. It's almost 8 times bigger than the rice genome, and it's much more complex. And what's been exciting about that project is I've had a collaborator for almost 20 years when I've been working on potato. And what's really been great for me is that I stay involved in projects where that genome information that I can generate and those analyses I can do, I see the direct application to breeding improved potato, taking what we might want to call myths about potato as a plant and as a crop that have been around for like 100 years, and using genomics as like a forensic tool to understand what have breeders been doing in the last 100 years to make potato varieties, and sort of deconstructing that. So that's actually been a very exciting collaboration with the potato community, because it is a very small research community, yet it's a very important crop. And to be able to see how genomics has totally transformed that process of breeding and understanding what potato is going on in terms of growth and disease resistance. So I think that's another exciting one where I'm much more connected to how that's being used today, because I have several projects funded on potato genetics and genomics.
Oh, definitely. And I noticed that you've got the project called Potato 2.0, which I love the name, by the way.
Yeah.
But can you talk a little bit about where you're at in terms of the progress on that project?
So the idea of Potato 2.0, so first of all, an important thing to know about potato is what we call, it's a polyploid. So it has double the number of chromosomes as plants such as maize or tomato. So it has double the number of chromosomes, and that makes breeding very difficult. Potato is also what we call vegetatively propagated, right? So if you wanted to plant a potato, you would take one you bought at the grocery store and it would eventually sprout and you would go plant it. So you don't have true seed. And that has a whole bunch of implications, this what we call polyploidy, and also that it doesn't have true seed. And so some work done probably 2 decades ago by Shelley Jansky, she was at the Department of Agriculture in Madison, Wisconsin, and she started with this concept of what if we cut the number of chromosomes in potato in half and make it more like tomato? And then also make it what we would call inbred so that the genome's simpler. And then like maize today, all commercial maize that's grown across the U.S. is what we call F1 hybrids, where they took a mom and a dad that look very similar to each other. And when they make the offspring, the progeny, that F1, we would call it, it is bigger plant and it has more yield. And that's what we call heterosis. So her thought was, can we make potato like corn and make it what we would call inbred, that the mom and dad are have just diploid, like 2 copies of the chromosome, and then we'll make an F1 and it'll be bigger. And so that was her idea, and she started work on that. And then about 10 years ago, I started a project where we started to make what we will call the Potato 2.0 Foundation, and we did a lot of work on the genomics of that. And now that whole concept has now been adopted by a number of breeders in not just the US, but also in China and also in Europe, where let's simplify the potato genome. Let's make it easier to do breeding. And let's see if we can get to this property of called heterosis, where you can take 2 rather robust parents, put them together, and their offspring's even more robust. So that project is funded by the U.S. Department of Agriculture, and that's led by Jeff Endelman at Madison. And it involves a lot of other breeders plus my lab. And what we're working on is trying to get what we would call genome resources of some key breeding materials that they'll use for this Potato 2.0 project. And so it's very exciting that there's been a whole collection of people at all different levels interested in sort of revolutionizing this crop to make it easier to breed so that you could also breed things with more disease resistance or also yield.
Oh, well, this is a fascinating project, Robin, and thank you for sharing some of these exciting successes that you've had over the course of your career. And I know as a scientist, you stay quite busy. There's a lot to read. There's a lot to do. But we always encourage our listeners to take a moment for themselves to pick up a book and read for fun. So do you have a book that you've really enjoyed that you can recommend to our listeners, whether it's related to science or not?
I really haven't read much lately. I would say the last set of books that I read that I sort of really, really enjoyed was the 3-book series, The Girl with a Dragon Tattoo.
Oh, great. What did you like about those?
Well, I never read anything by this author because I believe this was his first set of books. But what I really liked was that the characters were really rich. They had a lot of in-depth aspects to them. And then it was sort of a complex plot involving the characters and their history and their interactions. So I really enjoyed those.
Excellent. Well, we will add those to our reading list on our website if our listeners haven't had a chance to pick them up yet. So thank you for that, Robin. And I know we talked about a lot of different aspects of your career, and I think there are still people out there who don't really understand what it's like to be a scientist, and people have this picture in their mind of what scientists are like. So we try to break some of the stereotypes that persist out there. on our show by talking to scientists about kind of the real experience and what it's really like to be a scientist. And I think there are these quirky traditions you experience and just funny and fond memories that you share with colleagues that show a human side of science that not everybody gets to see every day. So do you have an example of your own or a story of your own that really showcases this human side of science that you could share?
It's interesting. So I'm at an academic institution. I have graduate students. I have postdoctoral research fellows. And it's a bit like raising children in that they come here for training and what we would call career development because they eventually want a job that pays them a lot of money and they get to do something they have passion for. And so for me, it's a very interesting process. You bring someone into your lab and they have very limited knowledge, but they have passion and excitement, and you train them and you help them develop skills. And then when they're really, really good, just like when you have a family and your kid is 18, they leave the nest and they're gone. And so it's sort of this like birth, development, and then they're out in the environment on their own. And so it's this cycle of, okay, now I have new people and I train. I really enjoy training them. I like seeing them grow and blossom and develop. And then, whoop, they're gone. And then they're on to their own career. And so it's a bit like having a family in that you're always at a different stage of their development. And it's nice to visit when they now have their own job and they're well established in their career. And then you can look back at what was the training process like and what were some good times.
Definitely. I love to hear that you keep in touch with some of these people because you're absolutely right. They go on, they develop their own careers, but they sort of become these colleagues and collaborators and people in your field. And it's cool to see different scientists that you've trained develop and kind of achieve these exciting things in their own independent research careers. So I guess, how do you celebrate successes, whether they happen while the people are in the lab or maybe even afterwards?
So when they're in the lab, they have milestones. All the students will have a milestone. They'll have to take their comprehensive exam and pass. They'll need to get their papers published. And then they start to look for a job when they get close. And then they get their job and then they move on. And then once they're in their own career, they have their own milestones, which they have. And so we definitely celebrate those. And for example, tomorrow, one of my students has their dissertation defense. I'm sure he's going to do fine. And I'm sure he's going to find the job he wants when he completes. So I think there's always things going on with different people at different stages of their career trying to meet their own milestones.
Absolutely. So does your lab have a particular tradition for how you celebrate something like a successful thesis defense?
Pre-COVID, we would always take the student out for drinks to celebrate and chat. And then during COVID, that sort of came to a complete stop. But that's typically what we would do, is we would go out and celebrate both people in the lab, people in the committee, their friends. We would go out and sort of celebrate because it's a big milestone.
Absolutely. And it sounds like you've got a great lab environment there for your trainees. You're dedicated to their training and you're answering some really phenomenal questions with this group of people. So we know that in science, there are often things that limit what you can do, whether it's the amount of people in your lab, the amount of funding you have, or just the feasibility of some of these projects that you're trying to tackle. So Robin, if we took away all of the things that normally hold you back and you had all of the resources you could dream of, is there one particular question in science that you would want to answer most?
This is what I would do. So potato is a great plant. It's a fun plant, makes us tubers, great source of calorie, great source of nutrition. But one of its problems is its genome is like a genome on cancer. It has lots of mutations in it, lots of problems. I would like to go in and fix them all. I would like to go in and make the potato the best crop there is and fix a lot of these mutations so that we could get quicker to this potato 2.0 being a reality. That's sort of what I would like to do. And I think it's doable. It's just, it would take a lot of time and a lot of money.
Definitely. And I know we're sort of referring to potato in a general term here, but there are so many different species and types of potato. So is there one potato that you would start with?
So a fascinating story is here in the United States, if you went to a steakhouse, you would get this big long, and you ordered a baked potato, you would get this big oblong potato that had a rough skin on it. And there's a high chance that that would actually be this variety called russet Burbank. And you could buy that at any grocery store. And that's a very interesting cultivar because it was bred in 1870 by Luther Burbank, a very famous horticulturalist. And he bred it in 1870, and it was the most popular variety around the 1900s. but it was actually a smooth-skinned potato. It stored well, of course. There was no refrigeration, so it was very popular, but it was smooth. It didn't have that rough skin on it. And it turns out that there was some mutation that happened in the field to Burbank, and it got this rough skin, and that's what's called russeting, and that's called russet Burbank. So the variety that you're eating right now that actually is planted on 40% of all potato acreage in the United States is a variety that was first made in 1870 and then had a mutation in it. So what I want to know is, what was that mutation? Because whatever it is, it was very important, and it's made it into this very prominent variety that farmers still grow today because, think about it, it's over 150 years old, and it's still high yielding. It has disease resistance. It has very good storage quality traits. Because remember, all potatoes in North America are harvested in the summer or fall, and then they're kept in cold storage throughout the winter to provide us with potatoes all year round, and also to make French fries or potato chips. So it's a really good variety, and I'd like to know what happened around 1900 to make that mutation, to give it that rough skin, which is a very beneficial trait for harvesting and storage.
Oh, that is fascinating. Well, thank you for dreaming big with us on the show and giving me and our listeners something to think about today. And I know we've covered a lot of different information, a lot of topics here on our program today, but I'd love to end with advice. So, Robin, is there one piece of advice that somebody gave you at some point in your career that really helped you that you want to share with our listeners today?
I actually have two pieces of advice, if that's okay. And I would say, first of all, whatever you're going to do in your life, most of us are going to work, you should do something you're engaged in, because that's going to keep you healthy mentally and give you a lot of satisfaction. And what also is important is a lot of people that I train always are hesitant to apply for jobs because they feel they're not qualified or it's not maybe the best fit. And my advice is you can't turn down a job offer you don't have. So if you have interest in something, you should apply, because you never know where that's going to end up. And I think my career is a little bit of that. I never knew where I was going to end up, but each time it wound up being a great opportunity.
Definitely. I think this is such an important message to share, because I feel like students today sort of have this perception or this feeling or maybe this anxiety that they need to have everything planned out in advance. And for so many successful scientists I talked to, that just wasn't the case for them.
Exactly. And I think that's the norm now, is not to be working at the same place for your whole career. That's actually unusual now. And there's just a lot of opportunities. And I think COVID showed you can work remotely and you can change positions and change careers. And I think that you should be happy. You should try to be happy in your work, because that's a lot of your life.
Absolutely. Well, Robin, thank you so much for sharing that message. Is there any other last piece of advice or a last note of inspiration you would like to end our conversation on today?
No, just it's been a pleasure talking with you. And I hope people have got a better idea of what scientists are like. And I think we're rather normal people with just some curiosity.
Absolutely. Well, Robin, thank you so much for sharing your stories and your experiences with us today. Listeners, it's been wonderful to have you here as well. And we hope you'll join us on our next episode of People Behind the Science.